Hi, everyone. Thanks so much for joining today. My name is Adam Resnick. I'm from the Children's Hospital of Philadelphia. And it's been a great session today, following up on previous session. I had the privilege of co-moderating this session with Dr. Storm, who'll join us shortly. And we'd like to start with a video from Ethan Daniels, one of our really courageous warriors.
Hi everyone, I'm Rally Kid Ethan. I'm 15 years old and just started high school. I'd like to share a little bit of my cancer journey with you. It all started in the spring of 2017 when I noticed a hard lump pop up on the top of my head. Since it didn't hurt and I didn't do anything to injure myself, my mom decided to take me to the pediatrician. After seeing many specialists and having many tests and scans, we still needed answers. Well, we finally got those answers. It was 12:33 p.m. on a Monday. I was eating mac and cheese from a green bowl in the kitchen, and my friend Logan and I were watching YouTube. And that's when my mom got a phone call. She answered the phone and immediately went outside. I know my mom, and I could tell that she was stressed, and I even saw that she was crying. After a while on the phone, she finally came back in, and she sent Logan home. She sat me down and said, "Ethan, you have cancer." I was diagnosed with B-cell lymphoblastic lymphoma. It was the scariest day of my life. The next morning, I made my first visit to the hospital, the place that quickly became my second home. Within 24 hours of finding out I had cancer, I started chemo treatment. I wasn't even fully awake when they started the treatment, but my family and I knew there was no looking back. This was going to be my new normal. My treatment protocol consisted of 12 different chemotherapy drugs and high-dose steroids given through my port in my chest, directly into my spine, and orally. I had 28 nights in the hospital, 92 clinic visits, 84 port accesses with chemo, 142 blood draws, 27 spinal taps, 143 chemo shots, 3,422 chemo and steroid pills, 3 surgeries, 6 trips to the ER, 9 MRIs and CT scans, 4 bone marrow biopsies, 4 blood transfusions. In July, I completed 1,220 days of treatment. That's 40 months. All of middle school. I miss doing all the things I love most, like playing baseball, hanging out with my friends, and acting. Just being a regular kid, you know? At the end of the day, the worst part is that on the outside I appear to look normal, but on the inside I'm a bit of a mess. Cancer has physically taken a lot from me. Even though I've completed my chemo treatments, the side effects are still hanging around. I still feel tired and weak. I have a very hard time sleeping. My bones and muscles ache. I have neuropathy and can't feel my hands and feet. I currently have the resting heart rate of 100 beats per minute because the treatment I received weakened the heart. The average resting heart rate for someone my age is right around 75. Chemo brain is a real thing. Some days I have trouble focusing and my memory isn't great. I am beating cancer because of research. Research and collaboration are the keys to better treatments with fewer long-term side effects and ultimately a cure. That's exactly what the Rally Foundation does. Rally has dedicated their efforts to making my life, along with other kids' lives, longer and better. They are funding the best research and clinical trials out there. They have walked with us, hand in hand, through my long journey. They were there to help us navigate the scary stuff in the beginning and celebrated with me when I rang that bell to end treatment. I would also like to introduce you to my best girl, Chippa. She was a gift from the Rally Foundation. She came home during the hardest part of my treatment and never leaves my side. Her ability to sense what I'm feeling is amazing. Sometimes she could tell something wasn't quite right, even before I could. She's the best listener and is always willing to take a nap with me. You know the saying, man's best friend? It's so true. I can't imagine my life without her. I am so excited to be a tiny part of such a huge collaboration. I know the amazing things that the Rally Foundation and Canines and Kids Foundation have already accomplished. Bringing together the brightest minds in pediatric and canine care and research is going to change the lives of both 2- and 4-legged friends. And that is pretty epic.
That was a really amazing video. So often we see kids like Ethan just really speak and convey the message way beyond their years. So that was really great, and a great way to start off this session. We'll start off with Carl June and Nicola from here at Penn, but I wanted to remind the audience that please, if you have any questions or comments, include them in the question and answer portion below, and then at the end we'll have time to review those and answer them. But next we'll start with Carl June, which I suspect most of the audience does not need any introduction to. Really, for us, I'm here at the Children's Hospital of Philadelphia to have someone like Carl across the street leading the development of CAR T therapy at the University of Pennsylvania and Everson Cancer Center has been obviously a boon for both pediatric and adult cancers. And obviously that connection across the vet school with Nikki has been ever so much greater. Nikki, of course, leads the Cancer Moonshot coordinating center for the canine cancer immunotherapy trials. And so you can really see this interconnected space of really leading edge development across the canine and human elements for CAR T. So we'll start with those presentations.
Good afternoon. It gives me a great pleasure to present our ongoing work to develop CAR T cells that are universal, and this is a tag team approach. We've been working with Nikki Mason, who will follow my presentation at the Penn Vet School. My disclosures are listed here, and this work is supported by the NIH. So the goal of our project is to develop universal CAR T cells for leukemia and lymphoma initially. And I will go over our approach, which is to use human genome engineering to render the cells resistant to rejection. In this case, in particular with the dog, to overcome xenogenetic rejection mechanisms of human T cells. And the translational output from this work will be to use dogs as a model system, dogs with lymphomas, to test candidate human universal CAR T cells. So right now, CAR T cells are commercially available for leukemia and lymphoma in the United States, and they're made from autologous cells. Shortly, it's anticipated that they will also be available for myeloma, targeting the BCMA target antigen. So at this point, both CD19 and soon BCMA-specific CAR T cells will be in the practice of medicine, but they have significant the issue of our individual so-called N of 1 manufacturing of the cells is difficult, logistically very intensive. And then there's the vein-to-vein time of how long it takes from the cells when they're harvested from the patient to when they're infused. And then there's another emerging issue, is that in some patients the cells have poor proliferation. In some cases, this is due to senescence or replicative Hayflick limits. And in this case, then, the use of universal cells or third-party donor cells would be the only approach so those patients could be treated. So there are a number of differences between when one would use autologous cells versus universal cells, and they could either be T-cell receptor T cells or CAR T cells. Our initial approach is to make universal CAR T cells so that they're not MHC restricted. The patient-specific T cells are able to cure leukemia and lymphoma in a high percentage of patients. We don't know about off-the-shelf CAR T cells, but they would have multiple potential advantages, such as a single lot of cells could treat multiple patients, rather than having N of 1 manufacturing in real time, as with autologous cells. One could make a master cell bank, in addition, the business model of standard pharma models with centralized manufacturing, whereas with patient-specific CAR T cells, they need to be done beginning at the point of care and then with complex logistics. But there have been reports now that so-called donor or third-party cells have had efficacy, the first being a report from Great Ormond Street Hospital published in Science and Translational Medicine in 2017 and treating a few children with infantile leukemia. So the problem with third-party cells is that they trigger alloreactivity, which is in this case host versus graft immune reactions leading to rejection of the cells. This doesn't happen obviously when one uses autologous T cells. So with various genetic engineering approaches, we believe that both TCR-T cells and CAR T cells could be rendered non-immunogenic so that they would not be recognized by the host immune system. And there's various approaches to do that, and that's the subject of our project. And cells are rejected by various mechanisms. In the case of solid organ transplants, they can trigger rejection where the host T cells become primed through both direct and indirect antigen presentation, leading to organ destruction. In the case of bone marrow transplants or hematopoietic stem cell transplants, a major issue has been with graft versus host reaction, which is caused by the donor T cells killing host tissues. And this is managed with immunosuppressive drugs. And there's a unique issue that arises when T cells are transferred into recipients. And this is called the hybrid resistance. So if one takes, for instance, a parental bone marrow into F1 generation mice, the bone marrow is rejected initially by recipient or host natural killer cells. And this is due to the missing self issue that the NK cells recognize the lack of the appropriate restricting HLA alleles. And this was first described in 1964 and then has been worked out over the years, the various mechanisms of this hybrid resistance. On this slide, the mechanisms of hybrid resistance are in paper from Kanda et al. In the Journal of Immunology. They took BLAC6 and BALB/c mice, and then F1 BALB/c by BLAC6 mice, and infused T cells intravenously and followed them. What they found was a striking and rapid rejection and clearance of the cells if they were allogeneic, whereas the syngeneic cells survived essentially indefinitely in these mice. So if one would expect that this were T cell mediated clearance, it would not be so rapid. And in many nice mechanisms were looked at in this study. They showed that if they depleted natural killer cells from the mice with an anti-ACIL-GM antibody, then that prevented the T cell rejection and elimination. They also found out that this occurred in T cell deficient mice as another line of evidence that T cells were not involved in this elimination of the early elimination of these infused cells. So what can we do then to avoid this allo recognition by the host immune system? One approach is to get rid of the allogeneic T cell repertoire, and it's been repertoire can be alloreactive. And so by elimination of the T cell receptor, this could be prevented. And the mechanisms of allo recognition by host T cells are both initially the direct pathway, and both CD4 and CD8 cells of the host can respond to the donor T cells, and there can also be indirect presentation and recognition. So our approach now to make these next generation universal CAR T cells is to first eliminate the host alloreactivity where the T cells that are infused could cause graft versus host disease. And we also want to eliminate the recognition of T cells by the host, the so-called host versus graft reaction. And to do this, we're using triple editing with CRISPR-Cas9 technology to knock out beta-2 microglobulin, C2TA, which in the T cell receptor alpha locus, rendering the cells CD3, MHC class 1 and class 2 deficient. Our second approach is to render the cells resistant to NK cell lysis by eliminating missing self and stress cells. And I'll show some experiments using HLA-E SCT, which is single chain trimer. These human engineered cells, and in a large animal model with Mickey Mason in dogs with lymphomas, using human T cells that have a CD19 CAR and are edited as I have described. In order to eliminate T cell recognition by both the host and the donor infused T cells, we've used CRISPR-Cas9. We published results back in 2016 using, in this case, the beta chain T cell receptor and beta-2 microglobulin knockout, rendering cells MHC class 1 negative as well as CD3 negative. And these cells were completely have lost their allostimulatory effects when incubated with irradiated peripheral blood mononuclear cells. But the problem is when one makes cells MHC class 1 negative, they lose their inhibition of that prevents NK cell lysis, so that if one has loss of MHC class 1, they end up being killed through activating NK receptors that are triggered because of the missing self. And this is a likely consequence that we will face with our T cells that have CD3 and MHC class 1. Approach then is to use HLA-E. HLA-E is a class 1 MHC molecule in the same region as HLA-A, B, and C. HLA-E, though, is monomorphic. There are two alleles, just different by a single amino acid. And HLA-E triggers NK receptors. It triggers the NKG2A, which is an inhibitory receptor, and it also triggers NKG2C, which was expressed as a heterodimer with CD94. So our experimental approach then is to take K562 cells, which are susceptible to lysis by NK cells, and then engineer them, give gain of function mutations by overexpressing HLA-E variants in the K562 cells and testing them with effector NK cells that are grown with IL-2 and IL-21 and LCLs. And these NK cells are very late expressing a number of NK activating receptors. And this is very recent data from Jerry Linnett's lab, where he's shown that if HLA-E single chain trimer expression reduces a variety of E to T ratios with NK cells overexpressing the K562 cells overexpressing a single chain trimer, lysis is abrogated, and this occurs with a number of NK donors here. These are healthy donors to grow up the NK cells. So this looks very promising as a way to eliminate lysis. Our goal then is to test these universal CAR cells that are edited at the track locus beta-2 microglobulin, C2TA, and with an HLA-E single chain trimer in a canine trial with Nikki Mason at the Penn Vet School. And she'll describe progress in that area. So in summary, we've shown that donor T cells can be rendered resistant to host T cell elimination, but that they are susceptible to NK cell recognition, and that HLA-E single chain trimer provides potent protection from NK cell mediated lysis. And we hope during this next year to begin a clinical trial with the Mason lab at the Penn Vet School. So in summary, I've shown work from Gerald Linnett's lab, Avery Posey at the University of Pennsylvania, and funded by an NIH U54 grant. I'd like to thank you for your attention.
First of all, I'd like to start by thanking Ulrike and the Canines and Kids Research Symposium Committee for the invitation to present here today. Today I'm going to be talking about our experiences with CAR T-cell therapy in dogs with lymphoma, and I'm really going to focus on some of the challenges that we have seen to successful CAR T-cell therapy that appear to be shared between humans and dogs. And I think that underscores the value of pet dogs with spontaneous disease for testing next generation CAR T-cell therapies that are designed to overcome these barriers. So it seems like yesterday that this paper was published here on the left, but it was now 6 years ago, and this was one of the first papers demonstrating the follow-up of human patients treated with a CD19-41BB CAR. These patients had relapsed and refractory acute lymphoblastic leukemia, and this manuscript demonstrated these sort of durable, sustained remissions that a number of these patients were enjoying. And since that time, attention has turned to trying to understand the mechanisms behind failure of CD19-41BB CAR T-cell therapy patients. And you can see that was actually addressed in a recent review by Crystal Mackel's group in Nature Medicine last year, where she divided up patients according to reasons why they failed. So you can see 40% here show a sustained complete response, whereas 60% failed. And that was associated with 10% of patients with an inability to be able to manufacture an autologous CAR T-cell product, 10% demonstrated primary resistance to the CD19 CAR, and the remaining 40% relapsed either with antigen-negative disease, which was 30% with a 4-1BB CAR, or 10% relapsing with antigen-positive disease that was thought to be associated with loss of CAR T-cells, perhaps because of failure of fitness of those T-cells that were used to make the product, or immunological responses against the CAR. So given the impressive responses that have been seen using CAR-19 in human patients with B-cell malignancies, including B-cell non-Hodgkin's lymphoma, we felt that it would be valuable for the veterinary profession to develop CAR T-cell therapies that would be effective for dogs with B-cell malignancies, in particular B-cell non-Hodgkin's lymphoma. We also thought that because these dogs are immunologically competent and develop spontaneous tumors, they would represent a valuable model for evaluating CAR T-cell therapies and maybe next generation CAR T-cell therapies that could overcome some of the barriers that were seen in the human patients. And so this slide here just demonstrates our ability to generate canine CAR T cells. These are actually going after CD20 and show that those cells are indeed functional. So what we're showing here is that we've developed a CD20-28 Zeta CAR here on the left and CD20 BB Zeta CAR here on the right. These are fully canine. It is a murine single chain that targets the canine CD20, but the intracellular signaling domains and the leader sequences are canine. And so you can see here there's a lentivirally transduced CAR expressing 10% of these T cells express the CAR, and when they're co-stimulated then with CD20 positive targets, which are the canine B-cell lymphoma cell line CLBL1s, you see an enrichment of the CAR, increase in percentage of the CAR after several rounds of stimulation, and that then is reflected in the expansion of the CAR T cells, and you can see you get a log increase after 2 rounds of stimulation. And we see something similar with the 4-1BB Zeta CARs as well. And then just demonstrating here, when we look at both the CD20 targeted BB Zeta CAR and 28 Zeta CAR, and we co-culture those with irradiated canine GL1 cells. This is a canine B-cell leukemia cell line that has been engineered to express CD20. We see nice production of interferon gamma, whereas the parent GL1 line, we do not see any production of interferon gamma from the CAR T cells. And then here you can see evidence of cytotoxic activity against GL120 cells in both the CD20 BB Zeta CAR and the CD20-28 Zeta CAR. So what this tells us is that we can in fact generate functional canine CAR T cells. So I want to move on to demonstrate to you some of the comparable failures that we have seen with dogs with B-cell non-Hodgkin's lymphoma. And this is one patient, this was a 5-year-old boxer dog that came to us with stage 5 B-cell lymphoma. And you can see when we look at the compartments, both peripheral blood and lymph nodes here, at the time he was treated with an autologous CD20 BB Zeta CAR, you can see that there is a predominance of CD20 positive, CD79A positive cells in the peripheral blood. He was leukemic, and in the lymph node. Obviously, the dog comes off chemotherapy after CAR T cells, and we do see an increase initially in the predominance of these cells. And then, of course, by the time we get to day 50, you can start to see that the percentage of CD20 positive cells is decreasing. And that goes all the way through to day 138, when you now see a complete loss of CD20 positive cells. And we see that also reflected in the lymph node compartment as well. But what you will also see, of course, is that you have an emergence now of CD79A positive, CD20 negative cells. And so this is an indication that this patient has persistent disease that now has become target antigen negative. So when we look at that a little bit more carefully and actually are able to select CD20 negative and more CD20 positive cells in this dog and perform a simple semi-quantitative PCR for CD20, you can see in the negative cells here in lane 3, not only do we have evidence of a band at 891 base pairs, this is the canine CD20 sequence, but we also now see a band at 552 base pairs, and this represents a splice variant, as you can see here from sequencing. So post-treatment with a CD20 targeting CAR, this patient has actually spliced out exon 4 and exon 5, which essentially codes for the second extracellular loop of CD20, and it contains in green here the domain that CD20 CAR targets. And this is the reason why this patient has essentially relapsed with a CD20 negative tumor. So one of the ways that is being used to overcome that type of antigen negative relapse is to consider dual targeting. And obviously we see this in the human field as well, where they're looking at combining a CD19 targeting CAR with CD22. In our case, we have a CD20 CAR, and so we were interested in developing a CD19 CAR. And we did this utilizing a phage display approach. So this is actually work that was done in collaboration with Don Siegel's lab at Penn Med. The idea here is that we generate a canine single-chain phage display library, and we do that from leftover spleens that have been removed from therapeutic splenectomy from dogs with benign splenic conditions. The RNA and cDNA is taken from these cells, and then Don has devised a large set of primers that will actually amplify up all the immunoglobulin genes. So those are responsible for both the variable heavy and variable light chains, both lambda and kappa. And we are able then to amplify up the repertoire, the variable heavy and variable light repertoire within those B cells. And we've done that from 7 different patients and put those together. Those variable heavy and variable light chains can then be recombined utilizing a flexible linker to generate single chains that then can be expressed on the surface of M13 phage to generate a fully canine single chain phage display library. And that library can then be panned against canine antigens of interest, in this case, canine CD19. And of course, using phage display, the phenotype of that single chain is linked to its genotype, so it's then very easy to determine the nucleotide sequences of the variable heavy and variable light chains that encode for that single chain that binds CD19. So this is just showing a following panning of that canine single-chain phage display library against the extracellular domain of canine CD19. This is an ELISA demonstrating that after the 3rd and 4th round of panning, we now have polyclonal phage that identifies or binds to CD19. And when we take that and make monoclonal phage from that polyclonal population, we can identify unique single-chain binders to CD19, as you see in the bottom here. We can very readily then identify the nucleotide sequence of those single chains and generate canine CARs that then can be tested. And so this is just an example of how we've done that. Here we're utilizing Jurkat cells that have a GFP reporter in them. And so the concept here is that we can transduce these Jurkat cells with our canine CD19 CARs, and then we can co-culture them with target cells that express CD19, canine CD19. And if that CAR is signaling, we should then see those Jurkat cells expressing GFP in response to the target. And so when we utilize one of those single chains that we have selected in a canine CD19 CAR, this is the middle row here, and co-culture those with scaffold cells, K562 cells, that have been engineered to express CD19, you can see that they do indeed light up following co-culture with these cells, demonstrating that that canine CD19 CAR appears to signal and does so in an antigen-specific manner. And we can also see signaling in response to canine B cells, where of course we don't see that with an irrelevant CAR or with the non-transduced Jurkat cells. So in addition to seeing canine patients relapsing with antigen-negative disease, we have also seen a persistence or relapse with antigen positive disease. And so we believe that this is associated with loss of CAR T cells, or failure of their persistence. And this is demonstrated in this patient here, where we've been tracking CD24 and BB Zeta CAR T cells in a dog with B-cell non-Hodgkin's lymphoma. So we've been looking both in the peripheral blood and in one of the affected lymph nodes, and utilizing qPCR to track those cells. And you can see initially, following some preconditioning and then infusion of CAR T cell product, we see a small expansion that then appears to be lost. And then interestingly, as this patient progressed, we treated this patient at day 50 with rescue chemotherapy. And following that, we now see an expansion of these cells. We've actually seen that in several patients that have gone on to progress with antigen positive disease. Treating them with rescue chemotherapy has led to an expansion of the CAR T cells that we can identify here within the lymph node. But again, that is then lost despite ongoing rescue chemotherapy. And what we see in this patient is the formation of the canine anti-mouse antibody, so CAMA rather than HAMA. And we think that that is in part associated with the reason why these CAR T cells might fail. Now, this dog received preconditioning with 300 milligrams per meter squared of cyclophosphamide, which is sort of a standard dose that we give for CHOP-based chemotherapy. But taking our lead from some of the work in the human field, and certainly here by Cameron Turtle, demonstrating that sort of a more effective, more lymphodepleting preconditioning regime results in better CAR T cell persistence and clinical response, we increased the Cytoxan dose to 500 mg per meter squared in our next couple of patients, when we were able to demonstrate that this could then lead to a failure, or basically prevent the canine anti-mouse antibodies from forming. So the preconditioning regime certainly seems to be important, and increasing Cytoxan seems to prevent the anti-mouse antibodies that were forming. We have also seen more recently in treating a dog with a CD24 and BB CAR T cell evidence again of expansion after a 500 mg per meter squared Cytoxan preconditioning, and that's demonstrated here. So this time we're looking in the peripheral blood, in the blue, and in the red is the lymph node. And in this patient, pretty much immediately after we administer those CAR T cells, we see expansion of the CAR T cells in the peripheral blood. And interestingly, this correlates very nicely with an increase or a spike in body temperature. So you can see up here, this dog's temperature went to just shy of 106 degrees, which is quite warm. But treating just with intravenous fluids and some supportive care resulted in this temperature coming down, and that again coincided with a reduction and disappearance of CAR T cells from the peripheral circulation. So I think this perhaps would be early evidence of perhaps an early sort of cytokine storm, again not sustained and not serious in this patient. Nevertheless, what we do see is, despite this expansion of CAR T cells in the peripheral blood, the B cells in this patient continued to increase. This is after the preconditioning. They decreased. Treated with CAR, we see the increase, and we see then loss of any control that was achieved by that preconditioning chemotherapy, and the lymphoma comes back very quickly within days. So how can we make these CAR T cells more effective in the canine system? And one way of doing this is to try and arm these CARs. And we've been working with Onze Smollett from Dan Powell's lab at Penn to try to develop a way of using some sort of inducible system that could be safe and hopefully promote the function of these CAR T cells in the hostile tumor microenvironment, which may be responsible for their ineffectiveness, as I just demonstrated. So in this situation here, Onze has actually developed this inducible system. So here we have one gene that is expressed constitutively. In this case, it is the CAR, the CD24 and BB CAR. And then there's a second gene that actually is inducible. And in this first set of experiments, that's a GFP. So here we have canine T cells that are transduced with this CD20 CAR and with this GFP-inducible system. Here's the CAR, there's no stimulus, so it's not producing any GFP. When we stimulate these cells with a CD20 positive CLBL1 cell line, you can see now we start to get GFP expression, and this is the positive control with a cytokine cocktail. So this tends to suggest that this system is functional, and as we signal through the CAR in response to targets, we can actually turn on that second gene. So now what Onze has done has developed a second inducible gene, which essentially is going to be canine IL-12. And we have transduced canine T cells with the CD20-41BB CAR, where now following signaling through that CAR, we can actually get expression of canine IL-12. And this is the early data from those experiments that just demonstrate that both CARs, one with just a standard CAR, the other with this inducible system, can produce, this is actually interferon gamma, in response to stimulation with CD20 positive targets like CLBL1 and the GL120s. And now, hopefully this week, we will get data back demonstrating that there is IL-12 being produced in these cultures. Finally, I just want to sort of lead into Carl's talk, demonstrating that we too have seen issues with manufacturing in dogs that have advanced disease and are heavily pre-treated with chemotherapy. So in these instances, we see T cell populations that have low levels of expression of CD28. And we'll see this with aging, we'll see this with repeated antigenic stimulation. But when we then take those T cells and try to make a product by stimulating them with anti-canine CD3, CD28 beads, you can see that there's a complete failure of T cell expansion in these dogs, two dogs here with advanced lymphoma, and that's in comparison to T cells from a healthy dog, which expand very nicely. So there's a very clear evidence of some form of intrinsic T cell dysfunction that prevents us from making a product in these dogs. And this has been addressed to some extent by a Japanese group recently that were looking at expression of CD28 and some of the checkpoint molecules on CD4 and CD8 T cells from control dogs and dogs that had either histiocytic sarcoma or other tumors. And you can see here, although not statistically significant, they also have recognized a reduction in expression of CD28 on CD8 positive T cells, and they've recognized an increase in the expression of CTLA4 and PD1, shown here in the CD8 compartment, on these cells from patients that actually have tumors, including histiocytic sarcoma. So this is an indication, again, that we're starting to see some early evidence, at least phenotypically, of exhaustion in CD4 and CD8 T cells from dogs that have spontaneous tumors. And when we look at just 4 patients here, so early days here, and just look at the survival of those cells as a function of the dose of CAR T cells that these patients received, there's not really very much correlation there. But if we look at survival as a function of the ex vivo population doublings per day, that's basically the expansion of the autologous T cells as we're generating a CAR T cell product, we can see that there's a relatively nice correlation between if your product is doubling nicely, then you tend to survive longer. And so this is, obviously it's preliminary data, but it suggests the fitter your cells are, the more capable they are of expanding in ex vivo to generate the product, perhaps the more likely you are to respond. And so with that, I'm just going to leave it there. Many people to thank, many collaborations, and thank you as well to the funders of the projects, including NIH and the Parker Institute.
Well, that was really a great way to get started for this session. And thanks so much to both of you. We'll now turn to Renee Chambers from University of Alabama at Birmingham and Department of Neurosurgery. Like the other speakers, she's really been a leader in the field and founding member of the Comparative Brain Tumor Consortium at the Center for Cancer Research at NIH. And has also been leading the principal investigator of the canine immunotherapy trial, really, again, demonstrating the need for and leveraging collaborations across neuroscientists and clinicians in ways that really show the promise of collaborative discovery. So it's to Renee, and then I'll hand over my position back to Dr. Storm, who will take over, and I think who overlapped actually with Renee at Hopkins when they were both there.
Good morning. I'm Renee Chambers. I'm professor of neurosurgery at UAB in Birmingham, Alabama. I'm presenting our trial design and preliminary data from Canine, an immunotherapy clinical trial in pet dogs with gliomas. This trial is funded by the NCI as a Cancer Moonshot Initiative precision medicine project, and it is a collaboration between physicians and veterinarians at UAB and regional veterinary colleges. Our intent is to evaluate a combination immunotherapy in canine glioma and translate our findings and those of our studies in humans to treat both people and pets. Our aims include establishing the safety and tolerability of an oncolytic virus alone, and then in combination with a checkpoint inhibitor, characterizing the immune responses using immunohistochemistry, flow cytometry, and cytokine analysis, and correlating to outcomes, progression-free and overall survival. We hope that sequencing studies will give us insight into mutations conserved across species to serve as therapeutic targets in the future. These are the age-adjusted incident rates of primary malignant glioma in humans and in the dog. You can see the average life expectancy associated with the median age and other similarities between the species. The pet dog brain tumor model is scientifically and ethically sound. The tumors are sporadic or spontaneously occurring. The dogs have a much shorter lifespan than humans, accelerating results in clinical trials, and numerous similarities shown here. In 2005, the Canine Genome Project identified 99% of the canine genome. Over 19,000 canine genes have human orthologs. Almost all of the genetic alterations that influence cancer progression in humans have been identified in the dog. Finally, this model offers many advantages over genetically engineered mouse models, including periods of latency, sporadic development, complex biology, heterogeneity within the tumor, and perhaps most importantly, an intact immune system. Preclinical studies that advance to human clinical trials often fail at phase 1 or 2. We hope that this comparative oncology effort bridges a large translational gap between the orthotopic xenograft murine model, in which cells are either implanted or injected into the equivalent organ from which the cancer originated, and a complex immunocompetent human host. Our coordinating and data analysis center is at UAB. Veterinary sites where surgery and treatment is performed include Mississippi State, Georgia, Auburn, Purdue, and other veterinary surgery and specialty centers. In stage 1, surgery to resect the tumor is followed by infusion of the oncolytic virus into the tumor bed. HSV-MO32 is a very immunogenic virus. It can replicate in and destroy glioma tumor cells without affecting healthy brain cells. It also expresses IL-12, a cytokine known to stimulate expansion and reactivity of T helper lymphocytes. Importantly, based on work by Pavin and others, canine lymphocytes do respond to human IL-12. In stage 2, the virus treatment will be combined with a small molecule inhibitor, indoximod. There are currently 3 FDA-approved human trials at UAB and children's hospitals with 3 different viruses. Having concurrent human and canine trials offers real-time results and information that can be readily translatable. The HSV-MO32 is in a phase 1 trial of safety and tolerability in humans with recurrent, non-operable gliomas. At Children's, the HSV-G207 is used in children with gliomas. And also at UAB, the HSV-C134 with a CMV gene and homolog of an HSV gene inserted to reduce neurovirulence and still maintain efficacy is being trialed in adults with gliomas at UAB. As far back as 1994, we were trying to demonstrate the usefulness of oncolytic HSV and viruses in general in the treatment of brain tumors in humans, trying to develop a virus with increased selectivity for gliomas. In this case, we compared the attributes of two viruses, one with a deletion and another with a stop codon, both blocking the gamma 34.5 gene to reduce neurovirulence. The virus we're using in this canine trial, MO32, is a non-neurovirulent and very immunogenic virus, triggering toll-like receptors and expressing IL-12. Oral indoximab is a small molecule inhibitor with immunostimulatory effects on three main cell types. It increases proliferation of the effector CD8 positive T cells. It reprograms T regulatory cells to CD4 positive helper T cells, and it downregulates IDO expression in the dendritic cells. IDO, or indolamine 2,3 dioxygenase, is an enzyme that converts tryptophan to kynurenine. It's expressed by gliomas and results in immunosuppression by inhibiting T cells. High levels of IDO have been associated with a poor prognosis, and IDO blockade is expected to improve the efficacy of immunotherapy. In stage 1, the virus is injected into the resected tumor bed. This stimulates an immune response to the virus, and then through cross-epitope spread to a host of tumor antigens that are released as the tumor cell dies. In stage 2, stage 1 treatment is followed with 30 days of daily oral indoximab. Again, IDO is an enzyme that converts tryptophan to kynurenine, found in high levels in high-grade gliomas, and results in immunosuppression by inhibiting T cell differentiation and immune evasion by the tumor. We expect indoximab to work optimally after the immune response is triggered, amplified, and directed toward glioma cells. As a result of blocking IDO activity, we anticipate a sustained anti-tumor response, allowing sufficient time for the responding cells to find and kill most glioma cells without immunosuppressive mechanisms. The majority of patients enrolled in the study to date are diagnosed with oligodendroglioma. Based on histopathology, approximately 46% are low-grade and 54% are high-grade. 22 dogs have been treated with a median survival of 188 days. This is a significant increase from published median survival data for untreated cases of 60 days. We are analyzing data from stage 1 now to stratify outcomes based on many variables, including age at diagnosis, tumor grade and characteristics, such as tumor cell type, Ki67, T and B cell density, and the dose of virus, etc. With immunohistochemistry, we've identified tumor cells, of course, as the majority of cells. IBA1 positive microglia or resident macrophages constitute the predominant white blood cell. Neutrophils are the second most abundant subset. T and B cells are rare, indicating that the tumors are cold, that is, lacking lymphocytic infiltrate. And Ki67 expression appears to correlate with grade. We'll also analyze distribution of cells in the adjacent normal, peritumoral, and intratumoral regions. Tumor grade, low versus high, is based on parenchymal invasion, leptomeningeal infiltration, microvascular proliferation, necrosis, and mitotic figure counts. Flow cytometry, using antibodies validated for the dog, separates, identifies, and quantifies cell types. This is the gating scheme created by Dr. Foote, our veterinary pathologist here at UAB. Using antibodies, size, and granularity, we identify different lymphoid cells as lymphocytes, monocytes, or granulocytes. T cells are then further divided into helper, regulatory, or cytotoxic. One preliminary finding correlating with the human data is a reduction in total numbers of T cells. This is from the peripheral blood of canine patients, suggesting that the T cells are sequestered. This is presumed to be a significant component of immunologic tolerance to the tumor. The Luminex bead panel uses magnetic beads to bind to canine cytokines and chemokines in the peripheral blood. Cytokines are hormonal messengers produced by T cells, especially CD4 positive cells, and are responsible for most biological effects in the immune system. They can be functionally divided into two groups, pro-inflammatory, or Th1, and anti-inflammatory, or Th2. Both IL-2 and interferon gamma are indicators of cytotoxic Th1 effector cell activity. IL-4 is a Th2 type cytokine. Another interesting preliminary finding is an increase in IL-4 expression in the peripheral blood CD4 positive T cells without an increase in interferon gamma, indicating a potential bias toward the Th2 immune response. This increase in IL-4 has also been described in humans with brain tumors and is believed to be caused by the tumor itself. The reason this is important is that it provides an opportunity for intervention and more personalized treatments. The cytotoxic T lymphocyte, or CTL, response may be weak either because the cancer is insufficiently immunogenic or because a substantial Th2 or other component of the immune response downregulates the generation of CTLs. We plan to correlate changes in the T cells with tumor regression or progression using MR and CT imaging with RECIST data to determine prognostic significance. So the questions we've generated in this early stage of the project are as follows. We've just completed enrollment in stage 1. We continue to acquire data from those patients as we begin enrollment in stage 2. We will follow survivals and correlate with immune and tumor responses with tumor sequencing to follow. Questions worth investigating. Does IL-4 production by T cells have prognostic value? Is there a Th2 bias in the tumor microenvironment with tumor infiltrating T cells similar to what's reported in human glioma? Is this the result of an IL-12, IL-10 imbalance as in humans with high-grade gliomas? Studies in humans have shown patients with high-grade gliomas have decreased circulating IL-12 and increased IL-10, suggesting again that the tumors have a major systemic effect on the immune system. And if so, can this be corrected with HSV-1 IL-12 therapy using MO32? In conclusion, this is the first of its kind canine phase 1 clinical trial building on high quality preclinical safety and efficacy data to delineate the safety and tolerability of the virus with and without a checkpoint inhibitor in dogs with spontaneous gliomas. We hope to reveal the impact of therapy on tumors and the immune system and compare these results with data from human trials. Preliminary clinical results suggest that MO32 is safe with potential morbidity and mortality benefit. In summary, this is a novel immunotherapeutic approach using combination immunotherapies in pet dogs with malignant brain tumors, proving effective and safe with increased survival times. It offers a viable alternative for pet dogs to the current standard of care, that is, surgery alone or surgery with chemotherapy and radiation. It provides an opportunity to identify new targets and therapeutic agents. It further validates the pet dog as a model for human glioma and further supports bi-translational research to the benefit of both species. This is the larger group of investigators who have contributed to this effort through the Alabama Comparative Oncology Network and the Southeastern Comparative Oncology Network. And these are the active investigators on the current trial. We have a very nice website with more information if you or someone you know is interested, and I'm happy to provide updates anytime. We'll keep you posted with our results. Thank you very much.
All right, thanks, Renee, and thanks for Adam for pinch hitting for me for a few minutes. I also want to remind the audience, if you have a question, please put it in the Ask Question box. So our next presenter is Stephen Dow, who's a professor of immunology in the Department of Clinical Sciences at Colorado State, and a colleague of Doug Pham, who's a classmate of mine in high school, and Rebecca Packer, who we work with. Today, Dr. Dow is going to be talking to us about his work on immunotherapy approaches to canine brain tumors, and anxious to hear about what he has to say, and as we can continue to build on this work of comparative and translational work. So with that, we'll turn it over to Dr. Dow.
Hello, everybody. Thank you very much for the invitation to speak at the 2020 Pause for a Cure Symposium. Great meeting, wonderful work being presented. This will be a series of studies from our group looking at a new approach to brain tumor immunotherapy. And this will build on some of the lessons we learned from the osteosarcoma study, which Cheryl London and I will be covering elsewhere in this symposium. An overview of the rationale for why we're doing what we're doing, and then we'll look at results from and then move into our canine brain tumor studies. So the approach we've taken is to do two things to the tumor microenvironment in the brain. So first of all, we want to modify the immune suppressive environment. We want to deplete the immune suppressive population, primarily of macrophages, but potentially other myeloid cells. And then we also want to reduce the functionality of these immune suppressive cells. So not only block their recruitment, but also prevent them from secreting immune suppressive molecules. And we're doing that by targeting the CCR2 pathway, which is the receptor monocytes use to migrate. And then we're also using beta-adrenergic blockade to reduce the mobilization and function of myeloid-derived suppressor cells, which includes both monocytes and neutrophils. And then we're going to combine that with a tumor vaccine. This is a vaccine that's been designed to elicit immune responses against cancer stem cells, which we know from prior work are very important in the maintenance and spread of glioma and glioblastoma. So we're going to take the combined approach. We're going to use two drugs to modify the tumor microenvironment. We're going to use the angiotensin receptor blocking drug losartan, which we've evaluated extensively in the osteosarcoma studies of dogs. And then we're going to combine that with propranolol, a beta blocker, which recent studies from Elizabeth Rapaski's lab and from other groups have shown that beta blockade, beta-adrenergic blockade can significantly reduce the immune suppressive effects of myeloid cells and have indirect effects on angiogenesis and T cell function. And those two drugs will be combined in with the cancer stem cell targeted vaccine, which will be generated from tumor cell lines from dogs that have been cultured in a way to make them more immunogenic. So this tumor spheroid lysate protocol comes from protocols for growing neural stem cells. And what I'm showing you in this image is that compared to plastic adherent cells, when you take cells, tumor cells, and take away their substrate so they no longer stay down, put them in serum-free medium with defined growth factors, they rapidly round up and form these spheroid clusters. And the immune profile of these cultures, of these spheroids, is much different than from standard tissue cultures. So they become more immune stimulatory, they upregulate expression of cancer stem cell markers, which will then be incorporated into the vaccine. And the vaccine is prepared using lysates from these spheroid cultures. And the lysates are combined with an effective liposomal TLR adjuvant that my lab has worked on for a number of years. And in mouse studies, this spheroid culture vaccine becomes much more effective at suppressing tumor growth, as seen in this image here, than conventionally plastic-grown, plastic-adherent tumor cell antigens. And then to modify the tumor microenvironment, as I mentioned, we're going to use losartan. This is a drug that our group has found, and Dan Regan published a nice book on this last year. It has an off-target effect on CCR2 signaling. As you know, it was originally designed to be a drug that blocks the angiotensin receptor 1 for modifying or managing hypertension. But we've repurposed this drug now for CCR2 blockade. It's the one drug out of a screen that we've conducted where we can consistently see CCR2 blockade at levels that can be attained in serum or plasma by oral dosing of losartan. And then this effect actually a property of it appears to be a property of the whole family of angiotensin receptor blocking drugs. So not just losartan, but other drugs like telmisartan and olmesartan. And in mouse models, Dan's work showed that continuous dosing with losartan in mice would eventually lead to depletion of macrophages in the tumor, as shown by this F480 staining, and also reduced angiogenesis, which we would attribute to as a off-target effect, or a consequence rather of depleting macrophages. You will also deplete angiogenic factors. And then the beta blockade part of this comes, as I mentioned, from Elizabeth Rapaski's work, but long story short, excessive adrenergic signaling is known to be an immune suppressive in cancer and other chronic inflammatory conditions. And if you can block that adrenergic signaling with drugs or knocking out receptors, you can significantly reduce not only the mobilization of myeloid cells from the bone marrow, but you can also reduce their accumulation locally in tissues and tumor tissues, and also reduce their functions. You suppress the secretion of immune suppressive molecules. And in mouse tumor models, blocking beta adrenergic signaling with drugs such as propranolol will significantly reduce tumor growth and reduce the accumulation of these myeloid populations. So we reasoned that it would make sense to combine a drug that blocks the recruitment of molecules into tumors with a drug that also helps block mobilization, but also blocks the functionality of the cells, even if they do make it into the tumor. And here I've summarized some of the effects of beta adrenergic blockade. These are data from rodent models primarily, which blocks the mobilization. Also indirectly improves T cell function, because if you remove the immune suppressive effects of these myeloid cells, T cells start to work again, and then you indirectly inhibit tumor angiogenesis. And in mouse models in our lab, we've shown that the combination of losartan and propranolol appears to be uniquely effective in stimulating the recruitment of T cell infiltration into tumors. These are sub-Q models where we're able to monitor tumor growth and look at recruitment of particularly T cells, which is the cell we want to get to move into tumors. So we've evaluated this approach originally in a mouse model of a mouse glioma model. It's an induced model with a tumor planted into the brain. And what I'm showing you here are a quick overview of the studies. These are the treatment groups, so untreated mice treated with only losartan and propranolol, mice that are vaccinated weekly, and then mice that get the combination therapy. And we monitor the response by IVUS imaging, histology at the end, and we're also going to be interested in tumor growth and survival. So what comes out of these studies is that the combination of the vaccine with losartan and propranolol gives us better survival times. But it's important to also notice that losartan and propranolol by themselves are also active compared to the vaccine groups. So both groups are active. The combination appears to be additive compared to the untreated animals. And importantly, these studies also gave us the confidence that this was not a protocol that led to significant toxicity. And then mechanistically, in terms of what's going on in the tumor, what we see compared to when we compare control mice, the vaccine only, we do see a mild increase in T cells, the magenta-colored CD3 T cells. Losartan and propranolol, interestingly, recruits T cells primarily in the perivascular region, just at the margin of the tumor. And then when you combine the vaccine and losartan and propranolol, you see a significant increase in the numbers of infiltrating T cells, both within the tumor tissue and in the peritumoral margins. So it looks like this combination really does drive a significant T cell response. So we've taken what we learned from the mouse studies, and we've designed a trial in dogs with glioma. Glioma is a relatively common brain tumor in certain breed dogs, which we'll see in a second. It's typically managed with either surgery and or radiation therapy. There's really nothing that's particularly effective for this tumor, very similar to the situation in humans. Interestingly, the genetic profile of this tumor resembles fairly closely the profile of pediatric glioma. So for a variety of reasons, the dog is actually really interesting and useful model for studying glioma. In the trial that I'll describe, the biopsy was confirmed, the diagnosis was confirmed by biopsy, MRI. None of the animals in our trials had prior surgery or radiation therapy. We're going to administer continuously high-dose losartan and propranolol orally, so throughout the study and beyond. And then the vaccine is going to be comprised of canine spheroid lysates mixed with an adjuvant. The animals will go through an induction phase for 12 weeks, and then they'll stay on maintenance vaccine therapy. So study results to date. So 9 dogs have been enrolled and completed the initial trial, and made it out to 3 months. What we can, and I'll show you the data here in a second, but the treatment was in general well tolerated, no adverse events, really beyond grade 3. Most were mild, ones and twos. No hypotension, for example, minimal bradycardia, and no immunological side effects that we could discern. And then we've reopened a trial now where we're actually combining the immunotherapy with radiation therapy. And I won't show you the results of that trial. Only 3 animals have been enrolled, but we've already seen significant increases in overall survival time, with 1 animal making it to 3 years and then several animals making it beyond the 1 year. In the immunotherapy only trial, these are the study patients to date. You'll see lots of boxers and related breeds represented. It's a very common tumor in boxers. There's a genetic component to the tumor. So boxers and Bostons and related brachiocephalic breeds are very highly predisposed to this tumor. This is a summary then of the treated animals and the types of tumors that we're dealing with. Most are oligodendrogliomas, a couple of astrocytomas. These survival times, we don't really have good historical survival data to compare to, but in general, without treatment, it's typically a matter of a few months. It's a rapid progressing tumor, very difficult to manage. And these are the survival times that we see coming out of the study. Several animals, as you will note, are still alive out past the year. This is what the tumor response looks like when it works. So these are examples of 2 different patients in study. This is pre-treatment MRI, and then going out to 9 months. And you can see that there's a 30% reduction in this animal's tumor. This dog had a more dramatic regression. This is pre-treatment, then at 3 months, you can see very significant reduction in the tumor dimensions. And again, this is only with oral TME modifying drugs and the vaccine protocol. If you look at a waterfall plot then of lesion volumes, pre and post treatment, you'll see that there's a group of 5 animals where the tumors regress during treatment, and then in this group of 4 animals where the tumors actually progressed and did not respond. So it's still roughly a 50% rate of response, which is a significant and dramatic improvement over what we've been able to achieve previously with only immunotherapy or conventional therapy. So we're quite enthused about these results. They certainly indicate that this immunotherapy protocol with drugs that can be repurposed so readily can be readily used to generate trials without having to go through an extensive drug approval process. The vaccine is made from cell lines, so it does not require tumor biopsies or autologous cell lines. These are allogeneic cells that can be readily grown and expanded in culture. So to summarize the study results, and what we would conclude is that targeting both the tumor microenvironment and the T cells currently can enhance significantly immune efficacy. The protocol was well tolerated, no significant adverse events, or it appears to be very promising as a non-invasive, non-toxic immunotherapy for managing brain cancer. And then, not surprisingly, it's likely we will see much better results when the immunotherapy is combined with either radiation therapy or prior surgical debulking to reduce the overall tumor mass. And where we're moving next in these studies is looking at the combination, particularly with radiation therapy. So to acknowledge the work, the people who have done the work, the neurology group has been fantastic. Rebecca Packer and Aaron Rosenthal have been the clinical neurologists managing these patients, and then listed are also the folks in my group who are backing up this work in the laboratory. Our work was funded by the Shipley Foundation and also by NIH, Canine Health Foundation. So I appreciate your attention, and thank you very much for the opportunity to present our work to you this afternoon. Thank you very much.
And thanks, Stephen. We'll now move on to the tag team performance of Jeffrey Bryan and Wayne Carter. Dr. Bryan is the director of PET Imaging Center at the University of Missouri and director of the Tom and Betty Scott Endowed Program in Veterinary Oncology. Today, Dr. Bryan will start the lecture off by telling us about the immune response against osteosarcoma in the Elias immunotherapy protocol. And that'll be followed by Dr. Carter, who's CEO of TVAX Biomedical. TVAX is the human correlate of Elias, and he will talk to us about a human clinical trial in GBM.
With Dr. Wayne Carter of TVAX Biomedical to introduce the translational potential of the Elias immunotherapy approach that has demonstrated benefit in dogs with osteosarcoma. I did present those data earlier in the osteosarcoma lecture, so I will mention them only briefly in this discussion. I do have a conflict of interest being a member of the Scientific Advisory Board of Elias Animal Health. The Elias immunotherapy protocol is made up of critical components that, working together, appear to cause an immune response against cancer in patients with naturally occurring disease. Initially, surgery for amputation and collection of tumor materials is performed. These tumor materials are used to create an autologous adjuvanted vaccination that's administered intradermally. And following the intradermal administration, peripheral blood lymphocytes are collected through apheresis. Those lymphocytes are transported to Kansas City, to Elias Animal Health, where they are expanded and then activated ex vivo into much larger numbers, and then are returned to the hospital to be transfused into the patient as an active immunotherapy approach. This is followed by 5 injections of low-dose interleukin-2, and the patients in our study received no chemotherapy following that and were simply followed for clinical progress. Each step of this important Elias immunotherapy progress has basis in historical literature with well-documented immunological mechanisms. In 1984, one of the first studies of autologous vaccination was performed in a colon cancer model in mice. Colon cancer cells, or normal mucosal cells, were collected and they were adjuvanted with Bacillus calmette-guerin, or BCG. And then the mice were vaccinated against their own tumor cells. And you can see in this graph to the right that the tumor cells that were adjuvanted resulted in a much greater delayed type hypersensitivity cutaneous reaction than did the mucosal cells, likely because the tumor cells carry mutations that act as neoantigens and therefore are stimulatory to the immune system. When tumor cells and mucosal cells are administered with no adjuvant whatsoever, there is almost no delayed type hypersensitivity reaction at all in the skin of these mice. What's important to understand about this is that the same result has been shown with almost every type of tumor that has ever been tested. So even immunologically cold tumors can be caused to create a cell-mediated delayed type hypersensitivity reaction when they are adjuvanted and administered as vaccination. However, autologous vaccination alone has not been very effective in treating clinical cancer. At the same time, adoptive T cells were evaluated for therapy. And in the first of its kind study, T cells that were isolated from the spleen of mice were co-cultured with irradiated leukemia cells to recognize the abnormal mutated antigens or neoantigens in these leukemia cells. And when administered back into mice, they could cause the elimination of this leukemia cell line from the animals. In the graph at the right, you can see that each dot represents the death of a mouse at a particular day on the x-axis. When mice received enough cells, 5 × 10⁵ lymphocytes, the tumors were eliminated from the mice and none in the study died. And this study has been repeated in a number of different types of tumors, but was the first of its kind to recognize that tumor-specific activated T cells could indeed clear cancer from a mouse model. It was later discovered that these T cells did not have to be tumor-specifically activated, that if the T cells were exposed to the tumor neoantigens and then removed from draining lymph nodes from the mice themselves, they were expanded ex vivo and then activated with staph enterotoxins, that those T cells were now active enough to eliminate, in this case, a fibrosarcoma model of intracranial cancer in these mice. And what you can see in these graphs is that the cells that were activated with either staph enterotoxin A, staph enterotoxin C, or even staph enterotoxin E were more likely to be able to cause prolonged survival in the mice than those activated by other means or not activated at all. So this non-specific activation of previously educated anti-tumor T cells could also result in an anti-tumor reaction in these mice. This was then further extended to identify that autologous vaccination into the animal themselves that could activate the T cells, and then the T cells could be removed and activated non-specifically, that those could also eliminate even existing metastatic disease. In this case, a metastatic model of melanoma showed that mice that were vaccinated with at least 3 × 10⁷ irradiated tumor cells admixed with C. parvum as the adjuvant for the vaccine, and then given adoptive immunotherapy, could eliminate all of the metastatic lesions already present in the lungs, showing that even advanced disease could be managed by immunotherapy, particularly in the setting of autologous vaccination and activated T cells. And in each of these cases, the immune mechanisms were well described and were recapitulated in many studies following them. Each of these components of previously well-validated immunotherapy approaches in mouse models was then extended through Elias immunotherapy into the naturally occurring disease of osteosarcoma in the dog species. Each of these dogs received a vaccination prepared from their own tumor cells and adjuvanted. They then underwent apheresis to remove those educated T cells from their blood, which were expanded and activated ex vivo, and then they were transfused back into the patients who then received low-dose interleukin-2 therapy following, and then were followed simply for clinical outcome. None of them received chemotherapy. And what we learned from this study is these dogs had an extended disease-free interval that in some cases, even when they relapsed, resulted in further survival more than is typical for dogs who received chemotherapy alone. We also saw an extended survival average of 415 days median, with 5 dogs alive out of 14 enrolled in the study at the end of a 2-year period following this infusion. So each of these previous immunotherapy tenets was recapitulated in a clinical response in these dogs with osteosarcoma. And because it has previously been shown that nearly any tumor cell type can create a vaccination that can cause a T cell response, the results of this study are potentially generalizable into other diseases in dogs as well as other diseases in humans. And it's the strength of these data that is allowing this to carry forward into a clinical trial in humans in brain cancer that Dr. Wayne Carter will now discuss.
Thank you, Jeff. Jeff and I are tag teaming this presentation. So I also would like to thank the organizers and Enrique for the invitation to present today. I'm Dr. Wayne Carter, the CEO of TVAX Biomedical, which is the human health counterpart to Elias Animal Health. Obviously, you've already heard of Elias Animal Health and the efficacy they have in osteosarcoma. And for disclosure purposes, TVAX also owns about 30% of Elias Animal Health. So just to remind us again of the technology, the surgery removes the primary tumor. We isolate about 50 million cells from that tumor in our lab. We then irradiate the cells so they're no longer capable of growth or replication, combine with GM-CSF as an adjuvant, a little bit different than in the dog. And then after the second vaccination, we do a leukapheresis to collect the mononuclear white blood cells, then take those T cells outside of the body, activate them, expand them, and then infuse back about 50 billion activated effector T cells back into the patient. And then we follow that up with IL-2 injections to continue the in vivo activation and expansion of those T cells. So this is an autologous cancer vaccine enhanced adoptive T cell therapy. Our therapy actually consists of 2 different manufactured products, the vaccine and the T cells. We have efficacy in multiple cancers. We really believe this is a platform technology, but today I'm only going to talk about the brain cancer results. We have very few side effects with the therapy. You get some mild swelling associated with the vaccination site, which is what we want because we want you to have a robust immune response to the vaccine. And then about 1/3 of the patients will have a low-grade fever and a little malaise for about 24 to 48 hours when they receive their T cells back. So it's a much different safety profile than most of the treatments for cancer which are on the market today. And we've just recently received FDA Fast Track designation for our phase 2B study in GBM. So I'm going to talk about one other preclinical study to sort of take us into the brain cancer realm. And so this is work that Dr. Gary Wood did, the founder of TVAX and our CSO, about 30 years ago. This is in a very aggressive rat high-grade glioma model. It's an RT2 glioma. And the control rats die off very quickly with this model. Importantly, as you can see here, all of the rats that were treated with the adoptive T cell therapy survived. And this was really Gary Wood's aha moment and really recognizing that this therapy was really something special. And that's when he really started and embarked on a very long journey for TVAX. This is a Kaplan-Meier curve which shows historic controls for high-grade gliomas. And as you can see, unfortunately, their survival drops off very quickly. This study in particular with the immunotherapy was done in human recurrent high-grade gliomas. And so these patients already had multiple surgeries and chemo, radiation therapy, et cetera. And so they had suppressed immune responses. They were really, most of them, at the end of their life. And yet, as you can see, we were able to extend the survival in really all of those patients by over 2x. And importantly, at the bottom of this, you can see that we were able to significantly increase the duration of the survival in about 20% of the patients. And again, it's important to appreciate that this occurred obviously in these patients that had recurrent high-grade gliomas after they had received immunosuppressive chemo radiation therapy. And so when you're depending on the immune system to work properly, it's amazing, honestly, that we were able to get this kind of responsiveness from patients that had such suppressed immune responses. This is one patient in particular that had a complete response to the therapy. This patient, as you can see, has a very large glioma, which is present at baseline. You can see it in these 3 slices. 2 months after the completion of the immunotherapy, you can see that tumor has decreased significantly in size. And then 8 months later, you can see it's almost completely gone. Ultimately, this was a complete responder. They survived over 5 years and then were lost to follow-up. I know there's a lot of interest in pediatrics with this group, and so we have actually treated about 6 different pediatric patients. This is one that had, again, a GBM, a glioblastoma, and as you can see here, a very large tumor present in this child. This is before the immunotherapy, and then 3 months after the immunotherapy, you can again see that tumor has decreased significantly in size. Moving forward, we're going to be focusing on adult GBM. We have fast track designation for GBM in adults, and we also have orphan disease indication granted by the FDA. There is no effective treatment for a specific subset of GBMs, which are the MGMT negative GBMs. Those are the ones that do not respond to temozolomide, and those are the ones which we will be focusing on in this study. It'll basically be a 1-to-1 randomization receiving standard of care, which is the temozolomide and radiation, versus the standard of care plus our immunotherapy. We have also considered a pediatric arm to this, and certainly we would consider it if we receive adequate funding for that pediatric arm to the study. This is what the study will look like. Again, surgeries performed, enrollment after the surgery, because we have to confirm that it's a GBM and an MGMT negative. They then receive their first vaccination within about a week. We continue to inject GM-CSF into the vaccination site on 3 subsequent days to continue that dendritic cell activation at the vaccination site. They receive then the second vaccination and also the GM-CSF, and then we will collect the white blood cells again through a leukapheresis. The radiation temozolomide is administered in a 6-week time point after the leukapheresis. And so all of this priming of the T cells occurs before there's significant immunosuppression from the radiation of temozolomide. And then we will administer the activated effector T cells back to the patient after the radiation temozolomide again is done, so that we don't interfere with the function of those T cells. And then the IL-2 is given to continue that in vivo expansion. So there are 3 key differences of this study in comparison to studies that we've done in the past. So these immune cells are going to be generated from patients that have healthy immune systems, as was done with the canine osteosarcoma study. It's also important to appreciate that these patients are going to have minimal residual disease, which is going to be brought about by a combination of the surgery and the chemotherapy. The third thing is that the cancer neoantigen specific effector T cells will be delivered by adoptive transfer after the patients have received the chemotherapy, which will bring about a more immunostimulatory environment for those T cells to hopefully have a more favorable environment than the more typical immunosuppressive environment which exists inside a tumor. Our challenges for TVAX have been funding, and funding. For the past decade, we simply have not been able to put adequate funding together to complete our human clinical trials. And so accordingly, in around 2014, we were actually able to put enough money together to start an animal health company in the Kansas City area. And so Elias Animal Health was started. You now know the results of Elias Animal Health. They've obviously had a successful osteosarcoma study and are moving forward with a registration study. Those translational results have also obviously given us movement and impact to hopefully move forward again with a human clinical study. In that interim period of time, CAR Ts have now been approved, and so adoptive T-cell therapies are now more real to pharmaceutical companies, to payers. TILs, the tumor-infiltrating lymphocytes, another adoptive T-cell therapy, are now also moving forward. And we've now also received Fast Track designation. So hopefully we are getting close to being past the hump and really then starting our hopefully our registration study. So thank you again for the invitation, and I look forward to any questions.
Great. Well, so thanks to Jeff and Wayne. We'll now finish with Elias Sayour. He's an assistant professor of neurosurgery and pediatrics and PI of the RNA Engineering Laboratory in the Preston Wells Jr. Center for Brain Tumor and Immunotherapy program. He's also a member and active participant in the clinical trial network PNOC. And today he's going to talk to us about the cancer ecosystem and ways to develop pediatric cancer vaccines. So we'll let Elias take it away.
Good afternoon, everyone. I want to thank the organizers for inviting me to speak here today, specifically Canine and Kids and Pause for a Cure. I'm going to be talking about accelerating translation of pediatric cancer vaccines through comparative oncology. So in order to develop new therapies against cancer, we need a better understanding of what cancer is. And cancer is not just cells dividing indiscriminately. Cancer is an ecosystem. And in this ecosystem, you have cancer cells, you have blood vessels that are creating a network within these cancer cells, and you have other cell populations in the tumor microenvironment. And these populations involve immune cells like macrophages and K cells and T cells. But the immune system in the cancer ecosystem tends to recognize cancer as a friend and not a foe. And this regulation within the tumor microenvironment is not just confined to the cancer, but it's also affecting the systemic circulation. And so there is significant immunosuppression within the systemic circulation. There is immunoregulatory mechanisms that affect compartments like the bone marrow, the liver, the lymph nodes, the spleen. So cancer is not just a local disease, but it's a systemic disease associated with profound systemic immunosuppression. In order to overcome this, we've developed a new platform technology that can activate the immune system both systemically and in the tumor microenvironment. And to do this, we've developed nanoparticles. Nanoparticles that can take up the genetic information of a patient's individual tumor, and when injected into the bloodstream, these nanoparticles can elicit an immune response within the periphery in these organs and within the tumor microenvironment. And by making cancer look like a dangerous virus, we get activation in all of these areas. So to do this, we are making personalized vaccines. And from as little as 100 to 500 biopsy cancer cells, we can extract total RNA. From that RNA, we can make a cDNA library. And from that cDNA library, we can amplify copious amounts of messenger RNA that's personalized to that individual cancer. We then load the RNA material into a custom lipid nanoparticle. These lipid nanoparticles are biocompatible, and when they take up the RNA by electrostatic interactions, we form these multilamellar complexes. These multilamellar complexes can then be injected right into the bloodstream through intravenous administration, And we found that when we inject these into the systemic circulation, we get activation of all of these immune cells in the bone marrow, liver, lymph nodes, spleen. We get activation of immune cells in the tumor microenvironment. And within the tumor, we see a lot of immunoregulatory mechanisms turning into activated mechanisms. And lastly, we see traditional vaccine effects. Dendritic cells, or DCs, are master cells of the immune system. They're responsible for educating T cells. And those T cells are responsible for trafficking to the tumor and killing the thing that they've been educated against. So from an RNA-loaded nanoliposome, we can see significant systemic and intratumoral reprogramming, and this reprogramming unlocks the vaccine effects that we're inducing. There are existing mRNA vaccine designs. Certainly Moderna, CureVac, and BioNTech are pharmaceutical companies that are prioritizing development of mRNA. mRNA is not inherently stable on its own. Although it can be stabilized, often a liposome is used, a lipid nanoparticle is used to protect that material, that mRNA, from degradation. And that RNA can be confined in an RNA lipoplex in the center of that nanoparticle, or it can be confined to the outside of that lipid nanoparticle in the form of an anionic RNA lipoplex. What we're doing is we're layering RNA in a different manner. We're layering RNA into these lipid nanoparticles using electrostatic alterations of positive and negative charge. We have a positive liposome and we layer negatively charged RNA, followed by another positively charged liposome, and then followed by another layer of RNA, and so on and so forth. And these multilamellar complexes we're generating actually condense into teeny tiny particles that are 100 nanometers in size because of the electrostatic interactions between the positive and negative charge. And so in effect, these are like nano onions. And we believe these are much more immunogenic than other RNA-loaded liposomes or RNA-loaded nanoparticles because these nano onions have much more RNA inside of them, and RNA can be quite immunogenic through stimulation of pathogen recognition receptors like toll-like receptors. And we've tested these head-to-head. When we compare multilamellar RNA nanoparticles to anionic lipoplexes or RNA lipoplexes, we see significantly increased percentages of activated dendritic cells with our RNA nanoparticle formulation. We also see increases in antigen-specific T cells, and we see significantly greater anti-tumor efficacy. And so on the far right is a murine model of cancer. This is a metastatic lung tumor model, and we see that RNA nanoparticles elicit improvement in median and overall survival benefits relative to other formulations. We also see anti-tumor efficacy in several other models. We see anti-tumor efficacy in head and neck cancer models, melanoma models, sarcoma models, and other metastatic lung tumor models. And for pediatrics, we've also investigated these in the context of murine models for osteosarcoma. Osteosarcoma is the most common malignant bone tumor in children. And certainly these vaccines given once weekly ×3 elicited significant improvement in survival benefit. We see that within these tumors, we're activating the dendritic cells, these master cells of the immune system. And although not shown here, we see these dendritic cells activated in every organ we assess. We see these activated in lungs, in liver, in bone marrow, spleen, and lymph nodes. Within the tumor, we also see an increase in central memory T cells. And when we take T cells from mice that were vaccinated with RNA nanoparticles and we restimulate them with the osteosarcoma cells, we can see a memory recall response through the production of gamma interferon. We also see activation of innate gene signatures within the tumor microenvironment. after an RNA nanoparticle vaccine, in as little as 24 hours, if we were to harvest a tumor after a singular vaccine, we see increases in innate signatures for effector dendritic cells and interferon regulatory factors and interferon response genes. So we're essentially shifting these regulatory environments from a tolerant state to an activated state in as little as one vaccine. When we funnel down mechanistically into what is eliciting these responses, we see increases in type 1 interferons. Type 1 interferons are part of the canonic antiviral signature. And this signature is important for fighting and containing viral spread. Interferon alpha, though, is also important for inducing an adaptive response. And when we block interferon alpha using monoclonal antibodies, we can actually completely abrogate the anti-tumor efficacy seen with this vaccine. And when we deplete plasmacytoid dendritic cells, we can significantly attenuate this interferon signature. So mechanistically, these RNA nanoparticles work through type 1 interferons that are being released from plasmacytoid dendritic cells. So based on these data, we have translated these findings into canine studies. We've initiated a canine study for pet dogs with terminal gliomas, and we've initiated a canine study in pet dogs with osteosarcoma. These studies in pets are so important because we can now evaluate the safety of this vaccine in a much more relevant model for cancer. These are spontaneous tumors. We do not inject these dogs with cancers. These are spontaneous tumors in these animals that will claim their lives. And these patients are eligible through the owner's consent for these types of therapies, which will allow us not only to learn more about the safety, but also to learn about what dose works best and what are the blood markers that show response in these animals. And then we can use this information for a human clinical trial, not only to get approval through the Food and Drug Administration, but also to better design a human trial. And so in our first patients with terminal gliomas, we were able to confirm some of the biologic correlates that we have found in our mouse models. Namely, we see increases in interferon alpha, these type 1 interferons that are so important for vaccine response to this RNA nanoparticle vaccine. We see increases in interferon alpha in as little as 2 hours in these canines. And this was an earlier signature than when we saw this in mice. In mice, we saw this around 6 hours. So knowing that this is increased in dogs in as little as 2 hours will help us in our human trial in terms of knowing when to look. These patients do develop low-grade fevers within a few hours of this vaccine, which we believe is a signal that we are inducing good inflammation and initiating response against the tumor antigens that we are presenting using these RNA nanoparticles. These patients with terminal gliomas don't get any other therapy other than RNA nanoparticles. They get a biopsy that allows us to manufacture personalized vaccines, but they're not treated with any other therapy, and these tumors are not resected outside of a singular biopsy. We also see activation of dendritic cells and T cells in these canines. We see this activation in as little as a few hours. This is very, very important because one of the reasons vaccines don't work in the context of cancer is they take a long time to boost, to elicit response. So to see activation of dendritic cells and T cells in as little as a few hours is a very good sign for this platform. We also see, interestingly enough, within a few hours, marked decreases in peripheral blood mononuclear cells, both monocytes and lymphocytes. And we believe that this is representative of these cells activating and marginating two lymphoid organs for the purposes of antigen education of T cells. But most importantly is the response. And in our canines with terminal gliomas, we have seen smaller tumors after 3 vaccines given 1 week apart. And interestingly, in some of these patients that develop smaller tumors, there's actually increased swelling, and these scans are read as pseudoprogression. And we attribute this to actually an inflammatory response within the tumor that's causing that increased swelling. We've treated 5 canines with terminal gliomas thus far, and within these patients, we've seen, again, this is a small cohort of patients, but we have certainly seen improvement in what the median and mean survival outcomes are for this disease. In terms of our osteosarcoma trial in canines, we've treated 3 dogs so far, and we have one long-term survivor. Based on this data, collectively, we were able to submit an investigational new drug application to the FDA. And this has since been approved. And we are now in the process of initiating a human clinical trial. And that trial is going to be in patients, adults and children with malignant brain tumors, and is expected to start in the next couple of months. And this trial really is only possible through the large animal work that we have done in pet dogs. And while this work has allowed us to reach human clinical trial application, it's also created a pipeline where we can do bench to bedside research and bedside to bench research, and using a large animal model as an inflection point in terms of allowing us to know whether it's safe to move forward into human clinical trials and whether things are working. So in summary, comparative oncology is an accelerant in translating discovery-driven efforts, and it has allowed for large animal studies in physiologically relevant systems. It has allowed us to identify human equivalent dose necessary for our clinical trials, validation of biomarkers, and the opportunity to help both of our pets and our children that suffer with these terrible diseases. I thank you for your attention and happy to take questions.
All right, I guess we'll open it up to any questions. I do see down in the new question box, if human glioma patients tend to be less responsive to treatment with increasing age, those in their 30s do much better than those in their 60s. Is there a similar situation in canines?
I can speak to that. We've not seen that to date, but we're still stratifying outcomes based on things such as age. So we may see that to date, we have not. With our 22 dogs, we've not seen a trend favoring older or younger dogs in survivals.
And then I think, Renee, in your data, it looked like a large number of the high-grade gliomas are oligodendrogliomas. Is that, are they 1p19q deletion oligos? I mean,
I can't tell you that yet. We've looked at the grade, the immunohistochemistry only, and so but we will tell you as soon as we get some sequencing.
Just because, as you know, I mean, oligos are quite rare, but it looks like in the canines, at least, I guess the histologic diagnosis is quite high. So I was just curious if you're finding the same mutation that we are in the human oligos.
Yeah, we're really looking forward to the genomic work, and we're sort of saving that for the end. We've archived tumor, and we'll do all the genomics at once. We've got nanostring, RNA-seq, and then whole exome plans, so we'll see.
I actually had a question again for any of the vets. When they talk about the brachycephalic dogs, like the boxers, In humans, Apert syndrome or Crouzon's are often brachycephalic. We know it's a FGFR receptor, but I mean, I can't remember ever treating an Apert's or Crouzon patient with a brain tumor. Yet it seems like it's the same. I think it's the same mutation in like pugs and bulldogs that we see in the Crouzon's and Apert's kids. I was just curious, is there any other known mutation along with the FGF2 receptor?
I don't know.
It's just striking, right? It's the same mutation, and it's all these dogs with that brachycephalic are getting these tumors, but we just in our craniofacial clinic we follow these kids well into young adulthood, and we just never see it.
It's true, and I will point out that boxers are predisposed to a lot of different tumors. So I'm going to guess that there is another driver in boxers that makes this multifactorial. The other thing is that pugs are a different lineage that don't come from the English bulldogs like the boxers and the Bostons do. Pugs are a similar mutation, separate origin, and so they don't get some of the tumors, particularly the heart-based tumors at the aortic. The aortic body tumors that we see in the Bostons and the boxers. So it'll be interesting to look at the difference in genomics between things that happen in pugs and the other brachycephalics, because that might be enlightening as to what we're looking for differences there.
Yeah, I mean, we're always looking, like you said, for those other predispositions. We can see a patient with Lee Fermini who's 7 and has a GBM, and her sister has the exact mutation who's in college and doesn't have a single tumor.
Right.
So there's obviously some other predisposition going on.
And probably epigenetic factors as well.
Sure. And then, Wayne, with the long-term survivors and that TVAX Kaplan-Meier curve, do you know any of those subtypes? Were they like IDH1 mutants, or was there anything that you knew when you looked at those long-term survivors that you were able to tell if there was a subtype or what mutation maybe?
Yeah, there's limited subtyping done. So we really can't draw any correlations there at all. Certainly, going forward, it's something we plan to look at more carefully, but not with the past studies.
Certainly exciting.
It is, and so I'm obviously a veterinarian, and so I appreciate the osteosarcoma data very well, and I also appreciate, I think, the translational aspect very well, and truly believe that it's a platform technology that can work potentially against any cancer. And I must reflect as I have listened to many of the courageous warriors and their presentations over the past 2 days. And I hear about the terrible side effects that they have, had to endure. And, I think that's one of the great things about, an adoptive cell therapy like ours is there are just so few side effects associated with it. And, I do hope that at some point treatments like that become more of the mainstay as opposed to starting with such aggressive chemotherapy and then the patients dealing with all of those side effects.
I see there's a question from Bryan from Hopkins. Maybe it was discussed earlier in a different session and I missed it, but if not, what is the status of checkpoint inhibitor development for canines? I think they will be needed to optimize T-cell inducing therapies. I don't know if Renee, you want to take that one?
Yeah, it's interesting. We've talked to several companies about trying like Pembro or different drugs. The Endoximod is just a small molecule inhibitor. I don't know, but I hope that the results of this trial will lead to sort of easier access, a willingness of these companies to provide drugs. This is no surprise to anyone. If you have a trial and you solicit a company to provide a drug, the last thing they want to see is a bad outcome from their drug that they're trying to market. So it's a little bit of you know, you need some pilot evidence to get them to participate, and you can't get pilot evidence without them participating. So hopefully with some results of these combination immunotherapies, these companies that are trying to get drugs, you know, sort of out there will be more willing to provide the checkpoint inhibitors. We are just now starting stage 2, so we haven't actually administered the checkpoint inhibitor yet.
Maybe, Jay, one thing that's worthwhile to mention, you know, is the sort of going live of the RACE Act this year. So the RACE Act is a congressional act really launched after an initiative by foundations and patient and families, where now pharmaceutical companies who are developing compounds or drugs for adult therapies are required to simultaneously assess those therapies in a pediatric oncological setting if the relevant target is available there. So, you know, it's one of the things that we hope, at least on the pediatric side, can provide for a common approach. But this could be, you know, really a great opportunity to intersect in the canine modeling space as a way to help prioritize and define such approaches across the pediatric and adult space. I'm not sure if any of you have thought about this or evaluated how, you know, canines can be a conduit for support in the pharma-based engagement and their need to potentially think across the pediatric and adult contexts.
It certainly seems, you know, like a reasonable thing. The challenge we have is sometimes the pharmacology of drugs is different in dogs than it is in people. And if Cheryl London were here, she would point out a good example of that is sunitinib, that results in some unstable metabolites in the dog, but tosarenib, which is a very close drug, is very stable and works well in dogs. And so assuming that we have good pharmacologic data and good PK understanding, then I do think that the dog offers an opportunity when the similar mutations are present, and certainly a number of those mutations have been identified.
I don't see any more questions here in the question box. We're a little over time, but if anyone has any pressing questions, we're happy to address them. Otherwise, I find this relationship between the canines and certainly children and all brain tumors unbelievably exciting. And for me, it's always great to hear what my veterinarian colleagues are doing, and we have a project going with Rebecca Packer that's going to be starting soon. So you know, it's neat to continue to work together and then hopefully continue to integrate. Especially what we can bring is the large whole genome sequencing to then continue to integrate that. As you all get more data, subtypes, and genomic sequencing for the canine. So I think it's a great partnership, and I always enjoy this conference and seeing everybody, catching up with Renee that I didn't see in years until last year's conference, and Nikki's like right across the street, and then you know, didn't even know all the neat stuff she was doing. So I said it's great for us, and you know, I know Adam and I are excited to keep moving forward. So thanks to all the presenters, great work, and look forward to staying in touch and keeping up again at the next conference. So thanks.
Take care.
Thanks, Jay.